DNA structure and replication. DNA structure discovery. Francis Crick and James Watson described the structure of DNA in a short article published in Nature on April 25, 1953, stating, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. The two DNA strands that are mirror images of each other due to the complementary bases provide the blueprint for making copies of the DNA sequence. The X-ray diffraction pattern of DNA provided by Rosalind Franklin and Morris Wilkins provided the key experimental evidence of the DNA double helix structure." Click here to see the letter Francis Crick wrote to his son about the structure of DNA. Nucleotides. Nucleic acids are composed of chains of nucleotides, monomers. Nucleotides are made up of a 5-carbon sugar, a nitrogenous base, and a phosphate. Nucleic acid. DNA. DNA is a nucleic acid made of 2 chains of nucleotides. In each chain, the nucleotides covalently bond together, forming a strong sugar-phosphate backbone, illustrated by the gray pentagons and blue circles. There are 4 types of nitrogenous bases, illustrated in red, purple, yellow, and green. The bases in one chain bond with the bases on the other chain with weak hydrogen bonds. The sugar is deoxyribose. DNA. DNA has 2 backbones that form the rails of a twisting ladder, a double helix. The bases form the parallel rungs of the ladder. DNA. The 4 DNA bases, abbreviated as follows: adenine, A, thymine, T, guanine, G, cytosine, C. Adenine only bonds with thymine, guanine only bonds with cytosine, so A bonds with T and G bonds with C. Complementary bases. The base pairs that bond with each other are called complementary bases. In DNA molecules, hydrogen bonds between complementary bases in the rungs of the ladder hold the backbones together and at a constant distance apart. DNA is antiparallel. Each strand has 2 ends, a 5' end and a 3' end. The 2 strands of DNA always run in opposite directions. They are antiparallel to each other. DNA is antiparallel. The prime notation indicates the number of the bond formed by the sugar's carbon atom starting from the bond with the nitrogenous base and ending in a clockwise direction with the phosphate group. The 3' position, OH, is the place where new complementary nucleotides will bond during replication. DNA replication. The functions of a cell are determined by its DNA. Cells have to reproduce many times. In complex organisms, trillions of copies are made from one original cell. But when cells reproduce, they must replicate or copy their DNA. The structure of DNA reveals how trillions of copies of the DNA in one of your cells can be made, and be almost exactly the same each time. DNA replication. The first step in replication is to separate the 2 DNA strands by breaking the hydrogen bonds between the bases, facilitated by the enzyme helicase. Each strand provides a template for creating a complementary strand of bases. DNA replication. DNA nucleotide monomers are made ahead of time and stored in the cell. They are attracted to the newly exposed complementary bases in the template DNA strands. The reactions are catalyzed by the enzyme DNA polymerase. The strands are joined together, resulting in the formation of 2 DNA molecules identical to the original. Many other enzymes are used during this process. Click here to watch a video about DNA replication. Semiconservative replication. The light green arrows show the direction that the bases are being added to each original strand, in opposite directions. Nucleotides can only be added to the OHN, the 3' of each original strand. The added nucleotides will bond with their 5' carbon to the 3' carbon location on the original strand. The new strands are built in the 5' to 3' direction. Since each DNA molecule has one strand from the original molecule, it is called semiconservative replication.